So let's start. Good afternoon and maybe good morning for some of you. And thank you for joining us. My name is Mar Markalev and I'm part of the team here at Levitas Bio. Welcome to today's webinar, From Tissue to Insight, Plant Multiomics powered by label-free cell enrichment. Before we start, a couple of quick housekeeping notes. Both of today's talks are pre-recorded, so they'll run smoothly, and we've saved plenty of time for live questions at the end. So please drop your questions into the chat as they come to you, and we'll get through as many as we can. The session is also being recorded, and we'll share it afterwards. The reason we wanted to put this session together is simple. Single cell and other omics have transformed plant biology and other fields. But for plant researchers, the hardest part often isn't the downstream workflow. It's getting the clean representative sample in the first place. So there's this bottleneck in the middle. Unlike animal cells, plant cells sit behind a rigid cell wall. So you have to remove that wall to release the protoplast. At the moment you do that, you are left with a fragile sample mixed in with a lot of debris, ruptured cells, free organelles, and in some tissues, you can have a great deal of starch. So you're trying to do something genuinely difficult, clean up that debris and boost viability, but at the same time, also trying not to bias your sample for any specific cell. And this is exactly the problem our technology was designed around. The idea behind the LeviCell is to enrich for life intact cells gently, no antibodies, no dyes, no barcodes, and without forcing the cells through the kind of stress that other sorting methods can introduce. Levitas Bio platforms are built on levitation, a concept originally invented by George Whiteside at the Harvard's lab, and then developed for biological applications by researchers at Stanford. The idea is simple. We apply a magnetic field that gently suspends or levitates your cells in solution, as you can see here. Because every cell settles at a different height based on its own density, the instrument naturally separates viable cells from dead cells and debris. And it does it all label-free, as I said, and in just a few minutes. The principle is straightforward. As I said, we suspend them in a paramagnetic medium and draw the sample into a channel, which you have a top magnet and a bottom magnet. That creates the magnetic field. And what the live cells are going to do are going to exclude the levitation agent and are going to levitate high. And the dead cells are going to include the levitation agent because their membrane is permeabilized and they are going to be more dense. We then simply collect the two fractions individually. And because the channel is pretty wide, 300 micrometers, can pass through comfortably. So if you have events that are big, they also will not clog the machine. And here's what that looks like in practice. So after levitation, you can find one of those images. This is a real levitation image from protoplasts. So you can see the top banded live protoplast on the one that is darker, and then all the debris and dead cells on the bottom. The yellow line separates the two fractions, so we can collect them individually. And you'll see this happen in real time in the movies our speakers are going to share later. And before I finish, one thing I want to highlight before we get into the applications is just how broad this approach is. Although a lot of work has been in mammalian systems, the same method works across a remarkable range of organisms, from human, mouse, zebrafish, and importantly for this audience, obviously protoplasts. And it spans a huge range of cell types and sizes, from tiny red blood cells right up structures like organoids. And the reason it generalizes so well comes down to three things, basically. Label-free, as I mentioned, it's gentle, so levitation keeps cells intact and alive, and it works at true single-cell resolution, so you can pull out even rare populations faithfully. So where does this fit into your work? Wherever you need a concentrated high viability suspension as your starting point, whether that's, for example, for single cell RNA-seq on platforms like 10x, flow-based analysis, or single cell metabolomics, the output will be ready, and you can go straight into those downstream applications. And it isn't limited to one sample at a time. We can scale up the workflow to high throughput up to 96 samples, so it fits multiple experimental designs. And rather than tell you more about it, I prefer that our two researchers invited today show you. And I'm going to introduce them to you. So our first speaker is Caroline Browns, a single cell application specialist at Wageningen University in the Netherlands. She earned her PhD at the Max Planck Institute for Plant Breeding Research in Cologne and went on to a postdoc at Umeå University in Sweden, working on plant hormone signaling and development. From 2020 to 2024, she managed the plant single cell platform at the BIB Ghent Center for Plant Systems Biology in Ghent, where she focused on evaluating and optimizing single cell technologies for plant samples, expertise she now brings to her current role. The work she's presenting today was recently published in an EMBO Press journal, where she explores different methods for preparing plant cells for single cell sequencing. Our second speaker is Sonke Viven from Professor Sarah O'Connor Department at the Max Planck Institute for Chemical Ecology in Jena. Sonke completed both his bachelor's and master's at the University of Göttingen, where he focused on plant lipidomics. Much of what he'll show you today builds on the pioneering single-cell metabolomics work of his PIs, Sarah O'Connor and Lorenzo Caputi, tracing how plants build complex, high-value natural products across different cell types. He'll walk us through his own work on single-cell metabolomics and why high-quality protoplasts are such critical as a starting point. And we are delighted to have you both here, so thank you so much. And without further ado, let's hand over the first one. 1 second.
Hello everyone, and thank you for joining me. My name is Caroline Krones, and I'm speaking from the Wageningen Single Cell Center. Today, I'd like to talk about the purification of debris-rich and large cell samples with a focus on plant single cell preparations and how we can make them more compatible with downstream transcriptomic workflows. Before I begin, I would like to acknowledge the team behind the data. This study was conducted at the VIB Center for Plant Systems Biology in Ghent under the lead of Professor Berthe Rebel and the team of the Plant Single Cell Platform. It was also supported by the VIB TechWatch Core, the VIB Flow Core, and the VIB Single Cell Core. So this project was very much a collaborative effort, bringing together the expertise from plant biology, cell handling, and also single cell technologies. To frame the problem of today's presentation, it is useful to first have a look at the main difference between an animal and a plant cell, which is that plant cells are connected by a rigid cell wall. Because of the cell wall, we first have to enzymatically get rid of it in order to release single cells, which we call protoplasts. This enzymatic digestion utilizes enzymes like cellulases, pectinases, and also hemicellulases to digest the primary cell wall components. But this necessary step also creates a trade-off. Once the wall is removed, the protoplasts become really highly fragile because the plasma membrane is now fully exposed. So although we gain access to single cells, we are also creating a very delicate sample that is very vulnerable. So during the preparation of single cells from plant tissue, we face recurring challenges, which are on the one side introduced by the cell wall digestion itself, due to the fact that now the sample does not only contain intact and alive cells, but also a high amount of debris coming from the cell wall material, but also ruptured cells, free swimming organelles, partially undigested tissue, as well as cell clumps that were formed after protoplasts have been released. And the other problem is that protoplasts are very large. They can get up to a size of 80 micrometers in diameter, and they are extremely fragile. So one has to be very sensitive with them in order to not rupture them or not induce additional stress. So it becomes quite obvious that sample purification is an absolute must in each of the workflows, but there are key requirements when it comes to plant single cell preparations, which is not only to be able to clean the sample up from the debris and to increase the cell viability, but also to maintain intact large cells as well as small cells, and to have all of this within a workflow that is suitable for downstream application for example, single-cell omics. There are alternatives for purification of plant samples, which include, for example, traditional washes or gradient steps. They are very customizable and easy to implement, which is a huge advantage, but they can prolong the workflow and also introduce more cell damage and loss in cell quantity. Another alternative is using fluorescence activated cell sorting or flow cytometry, which has the advantage that it's very customizable and also leads to a really high purity and precision in the sorting. However, it requires a lot of investment in order to get the technology, also to obtain expertise and to maintain the technology, and it can significantly prolong the workflow in itself. And it also still struggles with the cell size diversity that we have within the sample and the heavy amount of debris that is coming with the sample. The third alternative are label-free cell enrichment strategies, which are also referred to as gentle cell enrichment isolation strategies. These are more user-friendly in terms of that the platforms that they are using are less elaborative as, for example, from flow cytometers. They are also faster in the application compared to flow cytometry, and the conditions are a little bit more gentle to the sample in itself. And due to the possibility that the cells, even the ones that you are not interested in, can be reloaded, there's also no loss of the sample. However, the limitation is that there's no cell sorting capacity, so the precision in the sort is less than, for example, in flow cytometry. And today, I would like to introduce one example of such a label-free cell enrichment system, which is the LeviCell 1.0 system. This system is based on magnetic levitation for cell enrichment. So in the slides ahead, I will also use the term magnetic levitation by meaning the LeviCell system. So the principle is quite straightforward. First, the cells are suspended in a paramagnetic medium, and then the sample is poured into a separation track that is positioned between two opposing magnets. These magnets are there to create a magnetic field gradient, and then within the gradient, the cells mitigate and get an equilibrium position according to the density and the magnetic susceptibility. Finally, the targeted cells are enriched and collected into a positive and a negative fraction. And in the example shown below, you could already see that on the top part of the line, we are enriching for alive protoplasts, whereas everything below the line is mainly containing now root hairs, debris, as well as dead cells. The LeviCell workflow is relatively simple. It is one cartridge used per one run with approximately 250 microliter as sample input, which can be used one time only. The system produces two output vials, so one containing the alive protoplast, the second containing the bottom fraction that contains mainly the debris. An advantage of the system is that you can always separate the cells and debris again if you want to, so you can reload the not wanted fraction. And also important for the plant work is that there is no cell size restriction, because all of the channels that are used have a diameter of more than 300 micrometer. So the system is not as prone to, for example, high diversity in cell size as well as high or large cells, and also not as prone to clogging due to, for example, the high amount of debris that we have. There are adjustable enrichment parameters that you can choose on the device itself, which can, for example, help you in determining where the alive cells are that are stained in calcine in this case, or where the dead cells are, which are in my case stained with propylium iodide. And just to kind of like give you a very brief output. So when it comes to debris removal, the magnetic levitation is a real game changer, because in here I'm showing you an example of protoplasts isolated from roots in the top and also of leaf protoplasts. And you can clearly see that without purification, I still have a lot of debris as well as ruptured cells in both of these examples, which have been mainly reduced after the magnetic levitation has been performed. An additional advantage is that while you start with a larger cell input of something like 250 microliter, you actually enrich the cells that are intact in a suspension of roughly 40 to 60 microliter in the end when you use the Levicell, meaning that this is a high concentration mix of live cells which can directly be applied to single cell platforms without the need of further purification or further enrichment with, for example, centrifugation. We also tested the compatibility of the Levicells on different plant samples. So we see that there's a really broad tissue compatibility, but also species compatibility. I usually utilize a separation time of 5 to 15 minutes. And we have seen that this works really well for a diversity of species and tissues, and also a diversity of amount of cell debris in the sample. The cleanup fractions are suitable for downstream workflows. So, for example, for utilizing it for single-cell RNA-seq with 10x genomics or with the BD Rhapsody. And we also have already data published on some of these runs that we did with this, including the Levicell. We next wanted to benchmark the Levicell against flow cytometry devices that we usually used in our regular workflows. And for this, we loaded the exact same sample as input on 2 different sorters and the Levicell, and compared the cell viability after the purification to an unpurified input sample. So in both cases, the magnetic levitation as well as flow cytometers, we saw an enrichment of viable cells compared to the input sample. But we saw that the FACS gave a higher precision in the enrichment of viable cells. So if the only goal of the experiment is to maximize the precision in viable cell selection, the flow cytometry is probably a bit more precise. However, in our case, that is usually not the only criteria. And we therefore checked next the cell size distribution of the cells that have been purified. And here the distinction becomes quite striking, because the magnetic levitation was the only device capable of actually maintaining the cell size diversity in the survival or in the alive plants. You can see here that only in the unpurified sample and in the sample that has been purified with the Levicell, we actually observe the presence of larger cells, which is not the case for both of the flow cytometers that we tested. So this is very relevant for plant single-cell work, because there is most of the tissues, there is a developmental gradient present within the plant cell files. And this leads to the occurrence of, for example, smaller meristematic cells, but also larger, more differentiated cells within the same cell sample. If we are now losing the larger cells, it might be that we are also directly introducing a bias before even the sequencing begins. Of course, the purification is not the only useful outcome. So we also, at the next step, looked at the effect of the purification method on the transcriptome. Therefore, we compared the impact of the magnetic levitation and the flow cytometry on the cellular transcriptome by looking at single-cell RNA-seq data. When we are looking at overall transcriptome similarities, we do observe an influence of the purification method on the cellular gene expression pattern. But these influences can be cancelled out by, for example, using computational correction methods like batch effect correction tools. And this is important because it means that the purification step does leave a detectable signature, but not one that necessarily prevents meaningful downstream analysis. So instead, it becomes a factor that can actually be modeled and accounted for in the data processing. To conclude, the Levitas is now addressing a quite central bottleneck in plant single-cell preparation, in my opinion, which is the heavy debris in the sample that also contains unusually large and fragile cells. And the method provides a plant-ready cleanup, that it supports the gentle enrichment of intact cells, and it also maintains a suitability for transcriptomic readout. Compared with FACS, I think that it can be positioned as a practical and gentle alternative for large fragile plant single cells. And it may not replace FACS in every use case, especially when maximum precision is the priority, but it offers a strong solution when preserving size diversity, viability, and also biological representation. Thank you very much for your attention, and I'm happy to take questions.
Thank you so much, Carol, for that fantastic talk. And now we'll move straight to the other speaker, who is going to share with us his work with the single cell metabolomics.
As I was already introduced, my name is Sönke Beven. I'm a student in Sarah O'Connor's lab at the Max Planck for Chemical Ecology, and we are very interested in natural product pathways in plants. As most of you are probably aware, plants are the source to a broad range of high-valuable chemical compounds and drugs. And interestingly, the biosynthesis of many of these drugs is restricted to the plant kingdom. A big problem for an industrial point of view is that many plants only produce these high-value compounds at a very low yield, usually ranging from 0.01 to 0.1% of their dry weight. And if you look at these two examples of the anti-cancer medicines, vinblastine and Taxol here on the right, you can see that they are chemically very, very complex, and that makes the chemical synthesis in the lab usually very unfavorable. Therefore, it is very important that we understand how plants can produce these highly complex compounds to either improve their yield or even express the entire pathway in a non-host organism. In regard to understanding these pathways, the recent advancements in single-cell sequencing came in very handy for us. For example, in our model plant Catharanthus roseus, which produces the anti-cancer medicine vinblastine you saw on the previous slide, we were able to understand that the pathway is split over 3 different cell types. It starts in these IPOP cells, here shown in purple, then proceeds to the epidermal cells, and only the final biosynthesis happens in dedicated idioblasts that also store the final compounds. This kind of single-cell sequencing analysis is very helpful for plants like Catharanthus roseus, where we are already aware of most of the biosynthetic genes involved in the pathway. However, we are also working on different plants where often only the final product or very few precursors or very few genes involved in the pathway are known. In this case, single-cell sequencing alone can be very limiting for the pathway elucidation, as there's many cases in which right after the biosynthetic step we are already aware of, there's a transport event and the product ends up in a different cell type. We are therefore not able to find our next pathway gene based on the transcripts of the previously known cell type. There's also cases where right after the transport there's further conversion, and in very complex cases there's several rounds of conversions and transports. And for us it's very important to trace these transport and conversion steps for a biotechnological application, but also for us to look into the new cell types and then identify the next possible pathway genes in these new identified cell types. Due to these limitations and to not only rely on single-cell sequencing as an analysis, our lab developed a pipeline that allows tracing the biosynthetic pathway on a single-cell metabolomics scale. And for that, we start with probably the most important step of the pipeline, which is releasing all the cell types that might be involved in the pathway from the tissue by digesting with fungal enzymes. This is followed by the so-called cell trapping, in which we use our protoplast suspension and let them sediment into these honeycomb structures, these sieve wells. We can then use these sieve wells to put them into our dedicated cell picking robot. This robot has a microscopy setup which allows us to select single cells of interest and let the robot fully automated transfer these cells from the sieve well into a 96 well plate. This 96 well plate can then go directly into our high-resolution LC-MS system, which provides advantages over live cell imaging or spatial MS, as this allows additional resolution of the LC system in addition to the MS system, and then we can identify our compounds of interest and, in best case, identify new cell types. In our model plant Catharanthus roseus that was used to develop this pipeline, this was very successful. As you can see here on the right in the feature versus cell plot, you can see these blocks of metabolites localized to very distinct cells, representing dedicated cell types, also pathway. In addition to only tracing the biosynthesis over different cell types, this method also allows us to quantify the metabolites in the different cell types. In recent years, 2 colleagues from our lab took this to the next step, and we are now able to use the cell volume that we get from the cell picking robot and split it to 2 different plates, 8 microliter each, and we can now perform single-cell sequencing and metabolomics from identical cells. This kind of analysis took the understanding of the pathway to a complete new level, as for the first time we were able to correlate the transcription of known pathway genes and the metabolites. And this allowed us to identify a completely new cell type that only seems to store the pathway intermediate organic acid while not expressing any of the pathway genes we knew before. A big hurdle of this entire pipeline is that whatever kind of analysis for either metabolomics or multiplexing, we need high quality protoplasts to start the picking process. However, many plants that we are working with like to store their final compounds way and very difficult to access organs. This can be easy to isolate, and one very good example that we had is a plant that seems to have the entire biosynthesis located in rhizomes, and when we try to extract the protoplasts from these rhizomes, we do get protoplasts. However, as you can see on the picture on the right, there's a few protoplasts, as you can see, highlighted with red arrow, but mostly you see tons of starch. Our typical first step when we have this kind of problems and background contamination is to try the classical density gradient cleanup. However, in recent years we had several species where this was a big problem, because we saw that even within the plant there's a wide range of different sizes. We saw that different cells can have very different densities, and while some stay on top of the gradients, some sink down to the bottom or end up in some interface. And we also saw that some plants where the protoplasts are very susceptible to osmolarity, and for at least one plant we had a nice density gradient, but we couldn't recover the cells from the gradient because somehow the osmolarity was slightly higher in the interface where they were located, and as soon as we recovered them, they were all dying. Another big problem is that our mass spec system is set to a very high resolution and is therefore also very sensitive to background noise and, for example, to metabolites sticking to the starch granules. Luckily for us, we came across the Levicell system, and in this video that you should see right now, this is this high starch containing plant that we were never able to extract high quality protoplasts. And only with the Levicell you see this nice separation of starch granules at the bottom and some cell wall debris in the middle phase, and our protoplast at the top, and the blue line was in the separation line, and we were finally able to recover these nice high quality protoplasts. For our pipeline, the Levicell was really a game changer, as you can see on the right in the before-after pictures. Before, it all started in very little protoplast, and then in the bottom you can see these nice high-quality protoplast with only very little background. And then on the left you can see our metabolomics analysis, and on the very left there's this cell type that seems to only accumulate the late biosynthetic steps and none of the precursors, which is very interesting because we were not aware of this cell type before. To sum up my little presentation, our lab is interested in plant natural product biosynthesis, and to not only trace transcripts but also metabolites, we developed a single-cell metabolomics pipeline that allows us to identify where intermediates are produced, how they are transported, and where the final products are eventually stored. Since this pipeline is highly sensitive, we require high-quality protoplasts with very little background of a representative population of protoplasts. Some organs that produce these biosynthetic pathways can have contaminations in the protoplast due to either having high starch content or certain cell wall compositions that can make protoplasting very difficult. In this regard, Levicell was very helpful because it provides a very gentle method of purifying these protoplasts. Also, we found that it is less biased than other methods, and even if there's a bias and we can see cell types sedimenting during the purification step, we can see that on the camera and we can improve our method in this regard. And with this, I would like to thank you for listening to my presentation, and if there's any questions that I can help with, feel free to ask.
Well, and that brings us to the end of both presentations. A huge thank you to both of the speakers. Thank you so much, and for being here too, obviously, and for sharing your work and showing what's possible when you can start with clean, viable protoplasts. We'll move now to a short Q&A, and feel free to still drop your questions in the chat if you want, but we have some. We're going to start. This is a question for you both. So if you want to talk a little bit, well, where do you see plant single-cell sample preparation evolving over the next few years? One of you. Yes, Carla, thank you.
Go first. I think that it will become more and more interesting to actually find solutions on how to make better sample preparations. also because the application changes and also the diversity in species that we are working with will change, and diversity in tissues. So I think we have to come up with working protocols that are quite generally applicable, both in terms of species and tissues of interest, but also of application.
Thank you. Shanke, do you want to comment on that?
I absolutely agree that compared to mammalian cells, for example, we definitely have a problem more species and more varieties in the species and how the protoplasts look, how big the cells are, and how they behave to osmolarity and all these factors. And I think for us, getting a higher throughput, especially for the single cell metabolomics, is really crucial. But I hope that there's more technologies that you can find for mammalian cells, like pull down with surface receptors, will come down to plant cells at some point, and we have more ways to enrich very nice protoplasts.
Great, thank you. So both of you have used the Levicel. So there's a question about the user perspective. So how much training or expertise is needed to run the Levicel? So would you say it's accessible for new users? Both of you have used it, so you go ahead and tell us if it was difficult or not.
So for us, it was very easy. There's very few steps that we had to do. And after we got introduced to the machine here on site, it was very easy for us to teach new students and to get them running on the machines, because it's basically a handful of steps that you need to do to get good samples. And especially with the camera, it's very easy to track after which time your cells are suitable to basically harvest them. And that was very simple for us.
Yeah, I would agree with that. There is not really much training needed. The protocol is straightforward, can be implemented directly. And yeah, also the workflow is very easy to understand.
Caro, you mentioned working across a wide range of species and tissue types with the machine, with the Levicel. Were there any tissues where levitation required optimization or different conditions? Do you have a standard starting setup when approaching a new species?
Yeah, so I tend to restrict the levitation time to 15 minutes max. And then if there's no good separation, which depends on multiple kind of like factors, like for example, yeah, how much or how many cells I'm loading, and also what kind of like cell types I'm loading. I would either, yeah, continue or not, or maybe also rerun the levitation again on another chip. In general, I would say that this kind of like workflow is quite compatible with all of the species that I have tested so far. The only samples or tissues where I see that it's a bit tricky is or tissues where cells are in a very young meristematic state, like for example, in Schudapica meristems, for example, where cells just tend to stick closely or together just simply because of the pressure that has been generated in the tissue itself, but also because of the nature of the cell. So there I find it a bit tricky that cells are sticking together, so they form clumps, and they will of course then in any kind of like separation system either stay together as clumps or be compressed further and then form clumps again. You can kind of like prevent or you can still kind of run the output sample over cell strainers to try to declump the cells again, but it's not super successful. So these are the only kind of like types of tissues where I see that it might be a bit tricky.
Okay, thank you. And for Sanke, the combination of single cell RNA-seq and metabolomics from the same cells is really good. It's really exciting. What do you see as the main technical limitation at the moment? Sensitivity, cell numbers, or something else?
For us, it's definitely the cell number. I think in regard to sensitivity, especially for the sequencing part, we got pretty good. And also with the machine tuned to a very high sensitivity, we get a decent signal for most of the compounds that we're interested in, but a lot of the cell clustering and making the connection between metabolomics and transcriptomics, we really need higher cell numbers, as we see it for normal single cell transcriptomics. You need a good amount of cells to make any assumptions about the specific cell type and marker genes, and that's the same when we try to multiplex. And this is definitely a problem because both of these parts of the pipeline take some time to handle. And for us, the biggest problem is really increasing the number of cells that we can process.
Yeah. Well, thank you so much. I think with that, that will be all for today. Thank you to our two wonderful speakers. Thank you so much for your work and for also being here and having also this Q&A with us. And thank you also, everybody, all the attendees for joining us. If you'd like to learn more about how Levitas biotechnology can support your plant research, just let us know. And our team will be sure happy to chat about it and have a call. And have a great rest of your day. I hope it's not too hot. And see you at the next one. Thank you so much. Bye-bye.
Bye.
Thank you.